Our speaker tonight has his own Wikipedia page, which I aspire to have someday. Antonei Benjamin Csoka, Professor, Doctor. I've been calling you a lot of things today, but he said I can just call him Antonei, and I appreciate that. He's a biogerontologist at Howard University who works on the molecular biology of aging, regenerative medicine, and epigenetics. Csoka, Dr. Csoka has earned a bachelor's degree at Newcastle University in genetics. He has a master's in molecular pathology from the University of Leicester and a PhD in cellular and molecular biology from the University of Debrecen. He was a member of the consortium that identified the lamin A gene as the cause of the accelerated aging disease Hutchinson-Gilford progeria syndrome, and participated in the first National Institute of Health Progeria Research Foundation workshop. He also showed that progeria is a true representation of aging with respect to cellular signaling pathways and truly recapitulates the normal aging process at the cellular level. He currently researches the molecular etiology of aging at the level of signaling pathways. He has authored and co-authored over 50 scientific papers. Many of you know that Dr. Csoka is a proponent of life extension, cryonics, and transhumanism, and has been identified as one of the top 20 socially connected professors on Twitter. He is a scientific advisor to the Alcor Life Extension Foundation, the UK Cryonics and Cryopreservation Research Network, and the Lifeboat Foundation. How many people have heard about the Lifeboat Foundation? Check it out, check it out. It's really worth it. It's so cool. And a fellow of the Global Healthspan Policy Institute, and was featured in the first Immortality Institute film, Exploring Life Extension from 2005, produced by Bruce Klein. Let's welcome our good friend, Professor Dr. Antonei Benjamin Csoka.
So, yeah, thank you, everyone. Thank you, Neil. Thank you, Bill. Thank you, Janice. Really excited to be here. I'm going to be talking about different aspects of aging, beginning with the biology of progeria and then going into aging itself. And then in the latter part of the talk, I'll be going into like more of a deep dive into the philosophical aspects of aging. And it was kind of a nice coincidence. I didn't realize that you had Nikolai Fyodorov in your creed. I wasn't aware of that, and I will be talking about him and his philosophy towards the end of the talk. So as I said, it's divided into three sections, this talk. The first part is about my sort of the way I got into aging, studying it. And that, for me, that was through studying progeria. That was my entry point. And then that took me to cellular senescence, which I believe is what I call the causal nexus of aging. I'll explain what I mean by that. And then I'll segue into sort of broader aspects of aging, the theoretical aspect, which I believe strengthen the fact that progeria is truly aging. I'll talk about something called the Weissman barrier. I'll explain why I believe aging is what I call a meta-disease. So there's that sort of dilemma that you're probably aware of. Do we call aging a disease or not? I believe there's a solution to that by viewing it as a meta-disease. And then I'll delve briefly into what I think we can do about it currently. And then in the third part, I'll go into the philosophical implications, as I mentioned, touching on the simulation hypothesis and how that applies to our current situation. And also Fyodorov, Nikolai Fyodorov, and his hypothesis of the common task, or his directive of the common task, and how we can sort of merge the simulation hypothesis and the common task into like a broader mandate, perhaps. So first of all, this part one, my personal story of studying aging. So when I got into studying progeria, there was an idea around that can be put as a question, like, has nature given us clues as to what was causing aging? This was like the late 90s, early 2000s. So the idea was, can we learn something about aging from studying progeria and related disorders? And there's actually a whole class of diseases known as accelerated aging syndrome, which you see listed here. Hutchinson-Gilford is the one at the top. That's the one I studied, and that's the most well-known. It's the most severe form of progeria, and it basically dramatically accelerates aging. And the idea was, like, by studying these diseases, can we learn something about aging that we would never have thought of before? Like, is there something really unique there that would really just take us into a new dimension of understanding aging? That was the hope. And some people believe that. Some people believed, oh, it doesn't really have much to do with aging. There was a lot of different opinions. I believe now, I think it doesn't actually say, it doesn't tell us anything hugely unique. Like, there's nothing like truly that we would never have thought of. No, that's not the case. But on the other hand, I believe these diseases in Hutchinson-Gilford have taught us something very important as well. And that is sort of how I view science now, that you know, we all make hypotheses, we test them, and our hypothesis may not be confirmed. Like, we may not get the answer, but we still can gain a huge amount of knowledge doing those experiments, new knowledge. That's basically the basis of empirical science. So I believe that we did learn a lot from progeria. What did we learn? I'll answer that as we go forward. So this is a mother and child, and you can see that her son kind of looks in many ways older than she does. So this is a very typical presentation of progeria. Dramatic aging, like profoundly accelerated aging. And it's important to realize that this is not just something that looks like aging. So we're seeing heart disease, arthritis, joint dislocation, muscle loss, aged skin, hair loss. I mean, basically everything that we associate with aging, but at a dramatically accelerated rate. So children with progeria look like this at younger than 10 years old. I mean, it starts in infancy, basically. There's also some other things that don't appear to be related to aging, such as growth retardation. So they don't really grow. They don't really grow above several feet in height. And you also see strange things like recessed jaw. They can even reabsorb their tips of their fingers. They can lose their clavicles. You can see here, there's a resorption of the clavicle bones, and so on. And that was a real mystery as well, in addition to the aging, like what is actually going on with the tissue. I believe they're absolutely part of the same phenomenon. So they're caused by the same thing, the same reason. And that I believe is cellular senescence. So if your cells cannot divide, you will not grow. If your cells cannot divide, you will start to lose tissue from your fingers, from your clavicle. If your cells cannot divide, you will have delayed tooth formation, and so on and so forth. So these sort of features, these pathological findings that were thought to be not connected, the aging, are actually part of the same phenomenon, which is cellular senescence. So as we go through, I'll explain why I believe what we learned about aging from Progeria was not something that we would never have thought of, but rather it confirms that cellular senescence is hugely important for understanding aging. So I was involved in the identification of the gene, again, in the early 2000s for Progeria. And it was a long project. Francis Collins was the director of this project at the NIH. And I won't go into all of the details, but you can see at the top there a normal sequence of the gene that's mutated, lamin A, and excuse me, in Progeria, there's a single base change, C to T, you can see on the second line. And it's like that's one nucleotide out of the 3 billion in the human genome that was changed. So it was a C to T mutation. You can see that on panel B, that the top of the larger box, you see the normal sequence of lamin A, which is the gene that was mutated. And in Progeria, the line underneath, you see that C is turned to a T. And what that does is it doesn't actually, oddly enough, it doesn't change the protein sequence, but it changes the splicing of the gene. So on the lower panel where it says exon 11, that is exon 11 of the lamin A gene. And that nucleotide change causes the gene to splice in the middle of an exon 11, that it should not be doing. So it basically splices out 150 nucleotides from that exon. And that leads to 50 amino acids being deleted from the protein. So the net result is you have a protein that is being produced in Progeria that is missing 50 amino acids. And that has consequences for the cell. So the first thing we saw right away was that in cells from patients with Progeria, that you get this distortion in the nucleus. So this down here is like a normal human cell nucleus. This is stained for lamin A. So this on the bottom panel is the normal cell. The very bottom one is the normal cell. You can see it has like a nice round oval nucleus. The green is staining for lamin A, which is the protein that's mutated in Progeria. And the red is staining for DNA. And on the right, you see them merge together. So what we saw right away was that like this lamin A mutation, and the upper three rows are Progeria cell lines, that it causes this profound distortion in the nucleus. So you see this irregular shape and irregular nuclear membrane, which is you don't really see that in any other disease. Like it's a unique finding. So that was like, let me put this in context. This was very soon after identifying what the mutation was in the disease. So we wanted to test out how that might lead to aging, testing different things. So in the next few slides, I'll show you other types of damage that we detected in the cells. Understand, like, why, if we mutate this gene, this protein, do we get Progeria, accelerated aging? And various types of damage were found. So this is like a distortion in the nucleus. We also saw impaired DNA double-strand break. So the two columns on the left are normal cell lines. We irradiated them. And you see over a time course, so the upper panel, the upper row is unirradiated. The second row is at 30 minutes. The third row is at 12 hours, and the fourth row is at 24 hours. And you see that in the two normal cell lines, which are the two on the left, the DNA double-strand breaks, which are indicated by the purple speckling, are basically almost disappeared at 24 hours. So the bottom two lower left normal cell lines. Whereas in Progeria, you have this persistence of the DNA double-strand break And that's represented also on the graph on the right. So the progeria cells are basically not repairing their DNA double strand breaks efficiently at all. And there's even a kind of a basis DNA damage going on even before irradiation, as you can see in the upper right. So even prior to irradiation, there seemed to be some like low-level continuous DNA breaks being generated. If you compare the two on the upper right to the two on the upper left, which are the normal cells, you can see this accumulation of DNA break. So again, more damage. We also found that if we put progerin, which is the mutated form of lamin A, into normal cells, that we could accelerate telomere loss. So this is staining for telomeres, DNA specific to telomeres. The right is a control cell, the left is a progeria cell. We stain them with the telomere marker, put them through what's known as a FACS sorter. And it turns out that if you put the mutated lamin A into normal cells, you can actually accelerate telomere loss. So again, another form of damage, another form of DNA damage. This is staining for epigenetic changes. We see on the left a normal cell line. The upper left, is a normal control cell line, stained for lamin A in red. The green is a marker for what's known as heterochromatin. This means genes that are DNA that is compacted down and is not expressing genes. And you need a certain amount of heterochromatin in normal cells. Like you don't want all the genes being expressed. You don't want everything being activated. You need a certain amount of heterochromatin in normal cells. So you see that in the upper middle panel. There's a green staining there. And then the merging of the two is on the right. On the lower row, you see progeria. Again, you see the irregular nucleus that's always seen. But you also see a loss of heterochromatin, which what that means is genes are being expressed that should not be expressed. If you lose heterochromatin, you get something called euchromatin, which means the genes are being activated that should not normally be activated. So the message, what I'm getting at here, I'm not going into great detail with all of these, is that we just found various types of damage. Telomere, DNA double strand break, epigenetic changes. Also, another thing we found, gene pathway activation. So this was with Alex Zhavaronkov in silico. If you look at progeria cells, you look at the activation gene expression pathways, they resemble more old and middle-aged cells in terms of their gene activation pathways than they do resemble younger cells. Even the pathways are changed to an older pattern. So what does it all mean? What is it telling us? There's various types of accumulations of damage in these progeria cells. So we have, this is a normal cell here. Lamin A is usually lining the outer edge of the nucleus. So it laminates the cell. That's the origin of the term. And basically, it's causing all kinds of different damage inside the nucleus. It's changing gene expression and epigenetics, causing DNA repair defects. It's causing apoptosis, damaging telomeres. So at this point, it doesn't really tell us anything that we would never have thought of before. There's nothing like that jumps out, oh, we would never have imagined that. But what it does show is that, or I'll get to in a moment, is that the cells in progeria are becoming senescent very early. And you find that with all of the accelerated aging diseases. So in the earlier slide, I showed a list of different diseases, so-called accelerated aging diseases. And those are not caused by lamin A mutation. They can be caused by mutations in different genes, RECQ, DNA repair enzymes, and so on. So they have a completely different cause, a completely different etiology, but you get a similar outcome, like a similar phenotype at the end, the similar result, despite different mutation. What they all have in common is that in these diseases, you get dramatically accelerated cellular senescence. So you don't necessarily get exactly the same type of damage in each disease. But in all the cases, that damage leads to cellular senescence. And you're probably familiar with this idea of the Hayflick limit, that human cells or any mammalian cell will eventually senesce. So this is on the x-axis, we have the number of cell divisions. Y is proliferative capacity. Over time, the cells will lose their ability to divide. They will literally stop dividing. The only cases where that can be bypassed is in cancer. So some cancer cells can reactivate cell division. And that's what gives cancer cells their immortality, and that's what makes them so difficult to deal with. And the other case where you see this infinite replicative lifespan is in germ cells and embryonic cells. So they do have the ability to bypass this Hayflick limit. But the point here is that in the accelerated aging diseases, you get this dramatic decline in cell division, way more profound than you would see in a normal human being, greatly accelerated. And this is another experiment we did to prove that the progeria cells have increased levels of early senescence. This is staining for a marker. It's called senescence-associated beta-galactosidase. And you can see on the two cells on the left, the two cell lines on the left, staining for this marker of senescence, that you see it very early in the progeria cells. Whereas in the normal cells on the right, the two panels on the right, you do not see this marker. And this has been replicated many times. We know this is a fact. I've grown progeria cells in culture, and they literally stop growing very quickly. Like they're almost impossible to grow. You can grow them a few population doublings, and then they cease. And it's also been shown for other premature aging diseases. So the causes are, I want to emphasize this point. The causes are slightly different for each of these diseases, but the outcome is the same: senescence and dramatically accelerated aging. So this led to the conclusion for me that lamin A, the gene that is mutated in progeria, is regulating senescence. So you're basically starting out at the top there. You have this nuclear lamina disorganization, which is what the protein is doing. And then that causes all these other consequences that I've been talking about, some of which I've mentioned through those different experiments. But they all converge, they all lead to the same endpoint, which is senescence. And I was looking at this picture sometime later, after all of these experiments, with the knowledge of what we saw. And what struck me is, when I look at this picture, what I see in the child with progeria are visible signs of cellular senescence, like macroscopic representation of cellular senescence. So you can see a massive depletion of fat cells, adipocytes, loss of hair follicle stem cells. You see loss of muscle. You see loss of other tissues. You see loss of bone. Basically, all the tissues are being depleted. They're being destroyed because the cells are not replicating. Compare him to his mother. She has subcutaneous fat, subcutaneous adipocytes. She has hair follicle stem cells. Even things like hair graying are really caused by senescence, like you lose proliferation of the melanocytes in the hair follicle. So basically, this to me is like a visible representation of cellular senescence macroscopically. And it was remarkable for me to see that after sort of doing those experiments and taking in that knowledge. So that's what I believe is happening with progeria. That was the lesson for me. So there wasn't something, as I say, incredibly unique that we would never have thought of, but everything pointed to cellular senescence, which again wasn't exactly the answer we wanted, perhaps, but it's still like a profound insight. But what if we compare progeria to normal aging? In progeria, on the right there, it's caused by a mutation in a gene, lamin A mutation, in a gene called lamin A. That's the main risk factor, and that leads to all the things I was talking about: telomere loss, DNA double-strand breaks, epigenetic changes, stem cell depletion, cellular senescence, which also manifests as a lack of growth, lack of height, lack of tissues being maintained, resorption of bone, etc. In normal aging, we also see that, actually. So you do actually see loss of tissue, but it may be harder to see in some cases, so there could be, for example, like increased storage of lipids in adipocytes or something that could mask that effect, but it's still nevertheless there. That you see the telomere loss also in the cells, you see the DNA double-strand breaks, epigenetic changes, and so on. So the outcome is still converging on cellular senescence. Like, it's just that the cause in progeria is this accelerated damage coming from the mutation, whereas in normal aging, there's damage coming from a variety of different points. So it's this lamin A mutation that is accelerating all of the damage. So what it told me is that it led me to this conclusion that cellular senescence is the causal nexus of aging, is the term that I use. So what does that actually mean? A causal nexus means a bridge between cause and effect. So you can have multiple causes for something and one outcome, only one result, which is aging. But they need to go through a bridge, which in this case is senescence. So you can have telomere shortening, you can have mitochondrial dysfunction, aneuploidy, so all these kinds of different damage. But what they all do is cause cellular senescence, shown there. And that leads to a loss of proliferation in the dividing cells, a loss of maintenance of tissue. If you can't maintain the tissues in your body, you're aging. So they're basically synonymous with each other. Okay, you may say, well, there's some cells that don't divide, neurons, skeletal muscle cells, they don't divide. Well, they can also undergo senescence. They acquire something called the secretory phenotype, the senescence-associated secretory phenotype, also known as SASP. So they're not necessarily they're not losing proliferation because they weren't proliferating, but they still undergo changes. They still become senescent. So this is the term I came up with. The causal nexus is synonymous with senescence. Senescence is synonymous with the causal nexus. And basically, it's the bridge between all of this damage that's occurring, which is dramatically accelerated in progeria, but leads to the same outcome. So where do we put the mutation in this sort of causal chain? So if we think of all of these factors here, like mitochondrial damage, telomere erosion, cytoplasm damage, epigenetic changes, that central column there, all that is damage. That leads to the causal nexus, the intermediate step, which is senescence, loss of proliferation. And then that leads to aging, as we see on the right there. So where does the mutation fit in? It fits in at the beginning of the causal chain. So it's basically accelerating all of those types of damage, mitochondrial damage, telomere erosion, cytoplasmic, etc. So again, I coined this term supracausative. It's basically initiating the damage that is then leading to cellular senescence and then aging. So that was what I learned from progeria. I'm not saying that is the end of the story. There's still a lot to do there, but that's where, again, this is science. This is where my current understanding is of it. This is how I sort of hold that knowledge in my mind. I want to go now into like the broader support for that cellular senescence is the causal nexus of aging. Some theoretical support, and it basically goes all the way back to one of the pioneers of gerontology and evolutionary biology, August Weismann. And he was remarkably prescient. He came up with this idea, well over 100 years ago, that death takes place because a worn-out tissue cannot, these are his words, because a worn-out tissue cannot forever renew itself, and because a capacity for increase by means of cell division is not everlasting but finite. And he did this he came to this conclusion by studying single-celled organisms and comparing them to multicellular organisms and looking at how they divided resources between what are known as germ cells and somatic cells. So he was basically studying early life and looking at how those animals regulated cell division. And he reached this remarkable conclusion from that. He was also an evolutionary biologist. So he basically put this in the context of evolution. And Michael West has also sort of further developed this idea as well over the years, that essentially life on Earth has existed for billions of years. Like we all go back, we're all descendants of the first organisms on Earth. And the way that is maintained is through cell division. So this upper line, the green, is what's known as the germ line. So there's a certain type of cell that never loses the capacity to divide. I mentioned they will just keep going. And in human beings, those will be the spermatogonia and the ovarian ova, the ovarian stem cell. And they maintain life, not just from one human generation to the next, but basically over the entire evolutionary history of the Earth. Like those cells have maintained life. They are immortal, but our somatic cells are mortal. So it's almost like you can sort of think of a branch of a river coming off from the germline, which creates an individual. But those cells are destined, or used to be destined, to die. And it's sort of like you can imagine a tributary coming off and freezing. It doesn't have the properties to keep going. So you can see this in more detail on this figure. It's basically representing the embryonic cells at the top. So if you consider each column there one individual, so imagine this is one person. They started out as embryonic stem cells. And then early during development, some of those cells became germ cells. So this happens quite early in development. Germ cells, spermatogonia or ovarian stem cells will be produced early, and then they stay through the entire life of that individual. And then the majority of the cells, all the other cells, if you follow that column down, become somatic cells, somatic stem cells, and then just basically somatic cells. So those orange cells at the bottom are the majority of our cells. They are somatic. They have a limited lifespan. They will senesce. On that second column, you can see that red cell is senescent. And they're basically destined to senesce unless we change that. And but the point is also here that those germ cells on the second row the lighter green, can produce another generation, produce a new individual, which we see in the second column. So a whole new generation is produced from those germ cells at fertilization. And they will be embryonic cells. And then that individual will then make their own germ cells again, early in development. Their cells will become somatic and eventually senesce again. But then the cycle continues. They can at fertilization create a new individual, and so the life cycle continues. So there's no reason why cells can't be immortal, just that there are certain genes switched off in our somatic cells that render them mortal. And as I mentioned earlier, there is a way around that, which we see on the third column there, the cancer cell, and that has reactivated some of the embryonic genes. So they will reactivate telomerase, for example, reactivate other things, but it's not in a good way. So it's a kind of a corrupted form of immortality because you also have other mutations in the cells. So they can't be controlled, they can't be regulated. It is a form of immortality of the cell, but it's bad. It's not a good form of immortality. Basically the foundations of aging research were actually based on the idea of cell proliferation and cellular senescence. They may have called them different things. You know, Weissman may not have used the term senescence per se, but he was basically there already. And then Leonard Hayflick, proved that that was a real phenomenon. And then I think we have further proof through the accelerated aging disease. Switching over a little bit into like how we understand or how I view aging currently. So this is not directly related to Weissman, but if we are considering an individual, you can kind of stratify aging into different layers, different levels. So at the deepest level down here, we have molecular damage, damage to the DNA, damage to mitochondria, epigenetic changes, and so on. This is what you could call molecular damage. And this is really the cause of the higher levels. So you have all this damage occurring deep down, and then that's causing these other things. Obviously, that a big one is cellular senescence, that I consider the causal nexus. Then that leads to things that we normally associate with aging: chronic inflammation, nutrient sensing, deregulation, endocrine dysfunction. And then that leads to, in turn, to seemingly independent age-related diseases. So we see heart disease, we see cancer, we see Crohn's disease, we see all these different diseases that appear to be independent of each other, but they're really all coming from this basal level of molecular damage. So it's like the cause is occurring down here, the deep level in the DNA, in the mitochondria, in the organelles of the cells, and then it propagates upward through these apparently independent diseases. So that brings us to the question: is aging a disease? So is aging a disease? I don't think we can call it a disease exactly, because it's really the cause of the disease. So I think it's more helpful to classify it as a meta-disease, which is something transcending the individual diseases and the cause of these individual diseases. So what does meta mean? You probably heard that term before. Meta means like more comprehensive, transcending, or foundational, causal. So metaphysics is thought to be beyond physics, foundational to physics. It can be used to designate a newer related discipline, like metaphysics, as I mentioned. And I thought of this term meta disease. It came to me one day, and I looked it up, and apparently it has been used before by Abraham Maslow. He used the term meta disease to depict how the origins of physical disease are based on unresolved emotional issues. So it's not the same, it wasn't the same thing that I was thinking of, but in some ways it's similar in that he was looking for something basal disease, like beyond disease, that was causing disease. I thought of it in terms of aging. He was thinking of it in terms of emotional issues. But some of the principles are the same. So really, aging is the cause of age-related disease. It's not exactly a disease itself. Diseases, as we normally think of them, are various types of specific phenomena. Cancer, heart disease, arteriosclerosis, etc. So they're very specific. Aging is the cause of these things. And these causes, again, are occurring at the microscopic level. If we take cancer, for example, a p53 mutation is like very commonly found in different types of cancer. But it's not itself cancer. Like a p53 mutation is not cancer. But a p53 mutation can cause cancer. So it's like one is a cause, the other is an effect. Like aging is a cause, disease is the effect. So the mutation is the damage is the cause. The disease is the effect. So it doesn't quite make sense to call aging a disease, but it does kind of make sense to call it a meta disease. And you can sort of draw that out as a set in principle. Like this is a very simplified idea of this concept. You have this large set A, which is the meta disease, aging, which is caused by damage, and that is the cause of these different diseases. And then within that set, you have, say, set B, which would be cancer, and then within that set, different types of cancer. And then set C, you have, which is also a disease contained within the set of meta disease. You have heart disease. And you could even, if you wanted to, like include cosmetic changes like graying and skin changes and so on. So even those you could, in theory, contain within the meta disease set. It's actually Mikhail Blagosklonia, another aging researcher, said something similar along these lines. He said, a zoo contains many different types of animals, but it is not itself an animal. And I think we have to be careful, like how we define things. But I think it's a useful model, this set theory with meta disease as aging and then causing all of the other things. And it relates to something called linguistic relativity, also known as the Sapir-Whorf hypothesis. If we create new terms, it actually helps our thinking. It can sort of change if we create new words to define things, it can aid the way that we think of them. It can actually facilitate new ways of thinking about them. And this is an old idea known as linguistic relativity or Sapir-Whorf hypothesis, that the more words we have for something, the greater our perception of it. And there's like various examples. The Himba tribe have many different words for green, and it's been found that they have a remarkable perception of green color. Eskimos, the well-known example of the different types of words they have for snow, and this is a fact. Like they can perceive different types of snow. So it's I think as we go forward, not necessarily just in aging itself or even in biology, but it's always useful to try to make new words that help us think. And they're kind of sort of like putting a piton or like a wedge into a cliff face, like boom. this is a new word. We're going to define it. And, it's sort of like knowledge is sort of like climbing this cliff. Like we need new words to help us think. And it probably operates through synaptic plasticity. Like you create a new word, it probably causes some change in the. So what can we do about it? If it's up to me, I think it's wise to focus on cellular senescence. I think this is a smart thing to do. There's also, of course, epigenetic changes, which is a big thing now. I tend to think that cellular senescence is more important than epigenetic changes. And things like reprogramming are probably reversing cellular senescence. It's more important than reversing the disdifferentiated state of the cell. But I won't go into that deeply at this point, but I think I lean more towards cellular senescence as being primary rather than epigenetic changes. And in any case, like when we are thinking about defeating aging, I think if we can focus on something like cellular senescence, maybe epigenetic changes too, but one or two, three possibly like major mechanisms, then we can sort of fly over the walls of the maze. Each of those twists and turns goes to a different disease. You could spend years, decades trying to understand an individual disease, if ever, whereas you could just jump over the walls and get out of the maze simply by refusing to cooperate with the maze. You make your own rules. You define how you're going to get out. So I think reprogramming is a good way to go about this. As I said, I see it more in terms of reversing senescence than epigenetic disdifferentiation. And we've done some experiments ourselves at Howard, basically reversing, reprogramming progeria cells. So the upper panel there is staining for embryonic genes. So these cells started off as fibroblasts, just as totally somatic cells. And we're staining them for NANOG and OCT4, which are embryonic. So those cells have been reprogrammed. And you can do that not just with normal cells, but with progeria, like even progeria cells can be reprogrammed. So I think reprogramming is a potentially very powerful technology. And again, I see it more as reversing senescence than epigenetic disdifferentiation, but obviously, epigenetics is important. I just haven't personally quite decided on it yet. However, we can reprogram. We can reprogram cells. That's becoming a well-developed technology, but that's not enough. Because we need to go further than that. And Bill is going to talk about this more, I believe. Basically, it needs to be applied in more powerful ways to regenerate organs. So we can reprogram, but we need to go up the scale. We can repair the damage. We can repair the molecular damage. We can lengthen telomeres. We can even eliminate cells with senolytics, which I won't go into. But basically, we can do all this, but we need to regenerate organs. It's not just enough to reprogram the cells. We need to go up to higher scales on this stratified system. So that's about it for the progeria and aging itself. In this last third, I want to go into the philosophical implications of what this means to me, what I think it means for humanity. I want to start with the simulation hypothesis. So you probably all heard of this now. Elon Musk has been talking about it, various philosophers, Nick Bostrom and others have been talking about this for some time. And it's basically the idea that all reality is including the Earth and the universe, it's basically a simulation of some kind. So a technology that seems realistic to convince its inhabitants that it's real. used in movies like The Matrix and so on. I don't necessarily believe that we're in a computer simulation exactly. You know, I don't quite see it that way. But I think there may well be deeper aspects of reality beyond what we perceive with our five senses. So there could be like other aspects of reality that were imperceptible to us that are generating this material world. And if we consider it over time, you know, if we consider the universe over a very large time scale, so this time axis at the bottom there is going all the way back to the Big Bang and potentially even before that, like, you know, there must have been something metaphysical before the Big Bang. The ancient Greeks called it the arche, and there's like a lot of interesting philosophy there. But basically, what it shows us is that over time, the potential, you know, the metaphysical potential at the beginning of the universe was essentially infinite. So anything could happen, but nothing was yet actualized. Nothing had actually become reality. But over time, there's been a greater and greater conversion of that potential into actuality, into greater and greater complexity. And we are now at this point, as human beings, you know, perhaps somewhere in the middle of this whole journey, you know, where some of the potential has been converted into actuality. And according to some religious thinkers, Teilhard de Chardin and others, like you extrapolate this point way into the future, you arrive at the omega point, or God. And so you could look at this and say, well, as human beings, you know, maybe we have like a huge responsibility now to continue this, you know, this journey of conversion of potential into actuality, which is also known as entelechy. Like, can we contribute entelechy to the universe? And basically, each individual can, in theory, contribute their own development to the universe. So this is like, on the left here, we see Maslow's hierarchy. You know, we all have needs, survival needs at the bottom, safety needs. We need to belong. We have self-esteem needs. We have self-actualization needs. And everyone, you know, probably a lot of people have heard of this, Maslow's hierarchy. But he did, recently there's been the realization that he actually had a sixth level, which he called self-transcendence. And he didn't really talk about it much, like it was more in his private writings. But it's really, you can think of it as, you know, if we can self-transcend, we can actually contribute more to the development of the universe, which is shown on the right. So those same things I showed on the previous diagram are now shown as the universe's own kind of Maslow's hierarchy. And we can kind of like join ourselves to this self-actualization of the cosmos. But it takes kind of getting up to these higher levels, self-actualization and possibly self-transcendence, before we can do that. If we're stuck here, like in the lower levels, in survival needs, in safety need, we're never going to be able to really contribute to realization of the omega point. So, you know, humanity is kind of holding itself back in that way. You know, things like wars and conflicts and, you know, all kinds of disagreements and injustices, etc., are really causing individuals to be stuck, and that also leads to a potential sort of freezing of the development of life itself. So I think you know, when we do go about solving aging, we need to consider it in the context of other issues that humanity is facing. We can't just solve aging and just say, well, forget about everything else. Like, that could be potentially disastrous, I think. So we're also at this nexus point of a lot of other things occurring at the same time. Search for extraterrestrial intelligence. There's more and more data, information coming out about UAPs, and who knows where that's going to go. Artificial general intelligence, that's coming very soon. This is coming. How is that going to affect humanity? Exploration of the solar system. Elon Musk wants to go to Mars. I think that's a good thing. Want to go back to the moon. We need these things. But economic systems, maybe there can be improvements there. So it's like we need to, I think we can't solve these things in isolation. Like we need to consider them collectively and work on them together holistically. And in terms of the simulation itself, why would there be, you can ask the question, the rhetorical question, like what would be the purpose of the simulation? What would it be designed for? One potential answer is it's a test. It's a means to elevate consciousness. So within that diagram that I showed earlier, of the projection of the life of the universe, if we are in a simulation, there must be a reason for it. And one potential explanation is that it's a way to make life more complex, more conscious, to evolve life, to evolve consciousness over time. And it could be, this is getting quite speculative now, obviously. This is kind of very deep. But it could be, if the simulation wanted to design a problem that would require humanity to evolve, solving aging could be a way to do that as a means to elevate consciousness within the simulation. It would be a great challenge within the simulation, like the hero's journey collectively for humanity. You probably heard of Joseph Campbell's hero's journey, which is usually applied to individuals, but it could also be applied to humanity itself. Like this is our hero's journey. We have a call to adventure. We could refuse the call. I mean, certainly none of us have, which is great. We may meet mentors, but then we cross the threshold. We go into an unknown world, which is the lower part of that figure, the unknown. There's tests, there's allies, there's enemies. We approach a challenge, an ordeal, and then we may receive a reward of some kind. Maybe it's understanding aging. Then we have to go back. There's like a final test, resurrection, and then returning with the elixir. And this is like humanity change. So it could be applied to not just the individual, but humanity itself. And we're getting towards the end of this talk now, but this brings us to Nikolai Fyodorov, who is in your creed that we said at the beginning. He basically realized this. I think, like he basically realized this a long time ago. He was a founder of the Russian Cosmism movement, which was really a precursor to transhumanism. Yep, and what he believed in was radical life extension, physical immortality, resurrection of the dead, using scientific means. And he coined this term, the common task. That's what he called it. It's a creation of a universal brotherhood, and something that would unite humanity and to make us worthy to go forward into the universe, to govern the universe. And he saw like mortality, death, as the most obvious indicator that we were still not ready. We're imperfect. And he also saw it as the underlying reason for most evil and nihilism in the world. And he saw this as like a profound challenge that we had to address. But he also saw it as the way that it could unite all people of Earth, regardless of race, nationality, sex, gender, citizenship, wealth. Like it's the uniting principle of overcoming death, the common task. This is our common task. And I realized also that his ideas recapitulate alchemy. so alchemy was an older tradition that came before Fyodorov, but there were some similar goals. They were trying to transmute base metals into gold, but that's really an allegory for transmutation of consciousness from lower to higher level of consciousness. But they were also very much dedicated to creation of an elixir of immortality, creation of a universal panacea to cure all disease, purification of the human body and soul from the alchemical magnum opus. So it was really a very deep philosophical tradition. And it actually has some parallels with the common task. And the Philosopher's Stone, of course, was also connected with alchemy. But it really represents enlightenment. The Philosopher's Stone, it's not like a stone. It's not a physical object. It's kind of an allegory for enlightened mind or higher intelligence, which may also be now, we may have beginnings of contributions from artificial intelligence to that as well, in terms of the Philosopher's Stone. So in practical terms, what does this mean? What can we actually do? What does it mean for us here? One thing is sure, like only 1 or 2 generations will ever have the choice to fight for immortality. Because in the past, it was simply impossible. In the past, the alchemists were born at the wrong time. Fyodorov was born they saw it in principle. They saw what was coming, maybe. They knew that our time would come, but they were born at the wrong time. So no matter how smart they were, no matter what their ideas were, etc., it was beyond them. Alchemists, all the other greats. And in the future, it will be solved. In a couple of generations from now, there won't be any fight. There won't be any challenge. So it's a very unique challenge that will only ever come, even whether it's a simulation or not, it doesn't really matter. It will, this challenge will only be now. And we can expand this figure a little bit. There may be like a supercritical window of time in this, even within this narrow window, where the people that are born at the early stage of that time, of this window, have to do more. Like if they want to achieve physical immortality themselves, the individual, and this time scale is just illustrative. I put 50s, 60s on the left there. It could be different. I was actually discussing this with ChatGPT, and it came up with different. So, yeah. But, what I'm getting at is if those individuals here, for them to reach physical immortality, they have to do more than those individuals on the right. And the differences would be, for example, creating institutional changes, sociological changes, changing the world in different ways. Some of you here are doing that. But even people born later, they still have to make the choices they make will also determine whether they achieve physical immortality, lifestyle choices, development of technology, and so on. So there is some difference, but what is striking is success or failure could come down to a matter of months. So this was another thing I worked with ChatGPT on. Like you have individual A, born in 1970, they're healthy, avoiding risky behaviors, they survived to 2050. And everything is fine. You have the same individual, or similar individual born in 1970 and make similar choices, but they don't avoid a preventable health, like say they have some habit or something that causes some pathology to occur. They will not make it, and they, excuse me, these slides are out of sync. They will not, even though they're born at the same time and many aspects of their lives are similar, some failure of individual B to prevent a health issue could cause them to lose that grasping of physical immortality by a matter of months. So in any case, what we need to realize is that no single individual can solve this problem. Like we have to work together. It's something that can bring humanity together. It's the common task. This is something that you can unite us all. It is the great change, if you want to think of it that way. Again, whether it's a simulation or not, it can be the great change. And then the last slide I want to, it could also be, and this is speculative, this is, even more speculative than the previous slide. Say we don't do this now, like we don't find a way to do this, to erase our differences, to work together, to create harmony, using aging, defeating aging as the great cause, of the common task. Say we don't do it, and it could cause like a disconnection between the acceleration of technology and human consciousness. It could cause them to separate too much. And we're kind of at this bifurcation point where we could literally destroy civilization if we don't elevate our minds. And we could go either way. And maybe the common task is the way to do that.
Thank you very much. We're going over the Q&A time.